I'll start by thanking you all for being here today. I'm sorry, where are you? Let me know if you can't see anything. My thesis topic is on the effects of an antioxidant called Protandim on C. elegans senescens. And this research question stemmed from my personal interest in antioxidants and aging studies and the free radical theory on aging. But I came across, or my supervisor introduced Protandim to me, and he was interested. He was curious to learn if we could find a beneficial effect of the supplement on C. elegans lifespan. Could there also prove to be a therapeutic effect on his islet transplant patients? And we decided on C. elegans as our test subjects for many reasons, but probably the most important of which is the rapid life cycle. The wild-type strain lives about 2 to 3 weeks, but that would be irrelevant if they didn't share the same cellular machinery or similar genes to us. So all of this is important. Most interestingly, 60 to 80% of human genes have C. elegans homologs, and a large chunk of that are human disease genes. And that variation is dependent on the type of bioinformatic approach that you use.
I can change the slides if you want. If you tell me next.
So how can two organisms have the same number of genes, and you have 60 to 80% of human genes have C. elegans homologs, but 35% of C. elegans genes have human homologs, when they both have the same number of genes?
Because not exactly the same number. Are my numbers wrong?
No, you say you have almost the same number of genes, and 35% of C. elegans genes have human homologs.
I see what you're saying. Okay.
And 60 to 80% of human genes have. That's impossible, mathematically.
Do you know, I meant to delete this one because I wasn't sure about that. So I think I forgot to do that.
Okay.
Yeah,
This is the more accurate one.
Yeah, it sounds like a complement, right? Like maybe 30%. Like only 35% do not have a homolog.
Maybe
that's the only thing I'm kind of make.
Yeah, I think don't have human homologs. Maybe, yeah.
Yeah, there is. I'm sure there are certain genes we're just really not sure about, and that's why the bioinformatics approach you use matters. So I think I've come across different numbers, and it's a range, and so it's kind of ballpark.
Well, that number is going to be completely different now with the non-coding RNAs. When they get thrown in, it's going to be completely different. But in any case, I'm just saying that if you ever present...
Let's just pretend that this one's not here.
Yeah, if you ever present it someplace else, don't put both numbers.
Yeah.
Okay. I'm sorry, I don't even know how that is there, because I remember deleting it. That's the one I'm not really sure about. Okay, so...
we are in the show. I don't know if you want to go back to the final presentation, or that's the final.
It's the same.
Okay.
Yeah. They're both the same document.
Okay.
Is this the last one that I said?
Yep.
Okay. All right, so at any rate, the message that antioxidants are good for us is everywhere. It's hard to avoid. Many people subscribe to that message, including myself. And the idea behind taking antioxidants is based on the oxidative damage theory on aging, or the oxidative stress theory, or the free radical theory. It has a lot of names. And this theory claims that we have an accumulation of damage in our cells over time. More and more damage causes our cells to wear out, and then that leads to age-related diseases. The theory implicates free radicals as the driving force behind aging. However, it should be noted that loss of the catalytic antioxidant enzyme superoxide dismutase does not shorten C. elegans lifespan. And other studies on non-catalytic antioxidants have been done with mixed results.
Is that a morphology, like what kind of approach, superoxidative dismutase knockout C. elegans, or Transitory.
I don't know if they did RNAi or if they did no. I never looked at it.
So these other non-catalytic antioxidants, for example, vitamin E, 2 studies increased the lifespan and 1 did not. Likewise, Trolox, which is a vitamin E derivative, increased lifespan in 1 study, but not in another. Lifespan was also increased slightly by a mixture of tocotrienols, which are antioxidants similar to vitamin E.
Did those studies, the doses of the vitamin E was different, or do you know that? To explain the disparity between the studies?
I'm sure I would assume that they were. I don't have the exact dosages in front of me, but I would assume there were slight differences.
Well, assuming they were the same. But what do you think might account for these differences?
Well, I was going to say actually that probably dosage was a factor in the differences. I haven't gotten to that. But, yeah, alpha-lipoic acid increased lifespan in 2 studies, but N-acetylcysteine and vitamin C did not. And that reflects probably differences in the way that the compounds are administered, and including dosage. Now, what's really interesting about
What's not a possibility? I mean, if some compounds do and some compounds don't.
That the C. elegans didn't take it?
No, I mean, I'm sure. Let's say they took it up. I mean, they probably will.
Do you have a question? I'm not sure I understand.
No, so if you have some antioxidants that work, and some antioxidants that don't work, I shouldn't call them antioxidants because that's biasing you one way. Some compounds work, and some compounds don't work. One of the activities that's common among them is antioxidants, right? But they might have other activities.
Yeah, that's right.
They might have other functions in cells.
That's right, for sure.
So it might not be the antioxidant effect that these compounds are doing.
Actually, yes. So what I was going to mention is that what's interesting about the Trolox and alpha-lipoic acid studies was that the dosages that increased lifespan also increased heat shock. And that's interesting because it suggests that perhaps the result that in lifespan that we see is not necessarily via their antioxidant properties, but because it induces stress. On the contrary.
Which is the
Opposite.
complete opposite.
Yeah, exactly. I thought that was very fascinating. So there are some intriguing findings there with the non-catalytic antioxidants. Therefore, it can get a little blurry when we're looking at multiple biological mechanisms at work, interacting and overlapping. At any rate, the most persuasive tests of the oxidative damage theory are the ones that aim to manipulate reactive oxygen species by manipulating genes that encode the antioxidant defenses. Now, protandim is said to combat oxidative stress by activating the transcription factor NRF2, and this increases expression of antioxidant enzymes like superoxide dismutase and catalase. Clinical studies have been done in humans that showed an increase in red blood cell superoxide dismutase levels by 30% and catalase levels by 54% after 120 days supplementation. And the plasma was assayed for T-bars. T-bars is a widely used marker of lipid peroxidation. And it was found that T-bars declined an average of 40% after 30 days of supplementation, and the subjects were age range from 20 to 78. The ingredients in protandim are all herbal, 5 plant extracts, many of which are commonly recognized, for example, green tea. And turmeric is a spice that is used in a lot of curries. I have no idea what ashwagandha is, but it looks to be a root of some sort. And the other 2 ingredients are milk thistle and bacopa. Protandim is purported to work by being an NRF2 activator through a synergy between all of these ingredients. What do we know about NRF2? NRF2 is expressed in all tissues, highest levels being in the liver and kidneys, key detoxification organs. NRF2 may be further induced by cellular stressors such as heat, toxins, and radiation. In C. elegans, the NRF2 homologue is SKIN1, which is a known conserved stress response pathway. You might be thinking the more NRF2 the better, but actually overexpression of NRF2 is not always a good sign because it's been found that NRF2 was upregulated in pancreatic cancer cells and ductal adenocarcinomas. Although this could be a reflection of the cancer cell's innate ability to resist stress, rather than being a promoter of cancer, the NRF2 may be more of a protective mechanism at work in all cells. NRF2 also seems to have a mechanism that promotes atherosclerosis. For instance, NRF2 knockout mice had lower cholesterol levels in their blood and the liver, and they had a reduced expression of genes involved in synthesizing and storing fat and regulating glucose in the liver. So we can say that NRF2 plays a crucial role in many important processes. In our study, we used wild-type C. elegans, fed them OP50, and grew them on nematode growth medium plates at 20 degrees Celsius. I should mention that the study I'm about to describe was our second trial. We had a lot of issues in our first trial, the main one being that the protanum tincture kept seeping into the agar, and we didn't know how much. We had no way of knowing how much each of the worms were exposed to. We had no idea what the amounts ultimately were, and there was no observable difference in the worm's lifespan or any difference in phenotype in any of the concentration groups. So for that reason, we increased our highest concentration in our second trial to 3,000 micrograms per milliliter. And we added the protanum tincture not just to the feed solution, but also to the agar. We diluted OP50 with PBS, and we included an equivalent amount of ethanol in the control plates as in the highest concentration plate, because there is ethanol in the protanum tincture, and we wanted to include it in the control plate because it's known to affect C. elegans' lifespan adversely. The test worms that I used were taken from adult worms that were grown on the same concentration plates, and this was for the main purpose of increasing their exposure even from embryo stage. We tested 20 worms per plate, 2 plates for each worm, and I counted the worms alive and only transferred the live worms. If they still moved their heads or the tips of their head or tail slightly, I counted them alive. Even if they died when I placed them, by the time I placed them, they were dead. They were alive for that day. And there was this interesting burrowing habit I learned about from the first trial. I thought I had to watch for burrowing if there were scratches on the surface of the agar, but I one day saw a worm go down between the agar and the side of the plate. It was able to slip through there and go under, and it didn't come back out. I was shocked to see that the first time, and I thought it just made me question all my numbers from that first trial, and that's why I'm not reporting anything from first trial. But I learned from it, and so I accounted for it. And in the second trial, I would go back the next day. Up to 2 days later, I could still distinguish my test worms from the newer progeny. And by third or fourth day, then I would just use backup worms.
What are backup
Oh, I had some spare that were kept in the same condition.
On the same condition.
Yeah, because I expected that I could lose a worm here or there, and I wanted to be able to count for them all.
So you picked a live worm from your spare conditions.
Yes. Yeah, I know, that would be a factor.
That's biasing a bit, but that's okay.
But I didn't do that actually for many. It was only like a couple of times, so I don't think it would have affected, because if you see the numbers from second trial, I don't think it would have altered those stats in the end. Okay, so the results that were revealed, from our study, the C. elegans in the highest concentration plates showed very early signs of protandum absorption. I saw within just a couple days of them reaching adulthood, they had these really dark orangey yellow patches in the throat area. Eventually they had these dark patches throughout the length of their body. But it started, for some reason, it concentrated right here first in the highest concentration worms. And of course the C. elegans is transparent throughout its life cycle, so I was able to see that. In the end stages of life, all the worms appeared similar, except for this highest concentration group, which had that darkest yellowish hue, and I assume that's from the turmeric in the protandum, because I've had experience cooking with turmeric, and so I'm aware of its staining power. So I have pictures here. This was taken at about 2 weeks into the second trial, and I have all four concentrations, 0, 30, 300, and 3,000 plates there. You can see that these guys are still very much viable as compared to this plate. That's because these three concentrations, almost all of them are still alive at this point, 2 weeks, but almost all of these guys are dead. And this is like one of the few remaining live ones. You can tell that it's this cane shape here. He's not really able to move its body. It's able to move its head a little bit to eat. And this is another one here, but it's a dead one. And they don't naturally go in that shape, but I positioned them, I moved them that way just because it helped me to keep track when I was counting. That's a dead one. And you can also see from this image that it's very murky, this 3,000 microgram per milliliter plate. It was sometimes hard to see the worm even when it wasn't moving. And I have the lifespan results graphed here. The most and only notable difference you can clearly see is in the highest concentration group, which had much shorter average lifespans than all the other three concentrations, which showed no disparity really, and that's including the control in this three. The average lifespans of the three lowest concentration groups was about 19-20 days, in contrast to the 11-day average lifespan of the highest concentration group. That's like almost half. So you can tell that there's a statistical significance there without
This
even
would
running
make
the
sense to
numbers.
what you were saying before.
But I did run the numbers, and I was not surprised to see that to get a p-value of less than 0.003.
The cancer one?
Yeah.
So what can we conclude from this limited study? Unfortunately, not very much. Despite the drastic decline in that high concentration group of their life expectancy, many beneficial compounds and therapeutic substances can be toxic at a high enough dosage. So water, for example, you can drink too much of water. There's always limits. And 3,000 micrograms per milliliter is a very high dosage when you compare it to other antioxidant studies. In future studies, I think a better method to do this study would be to sterilize the C. elegans with a drug like FUDR and test them in liquid culture in microwell plates, because this would allow for a more rapid identification of substances that extend their lifespan, because you could test them on a much larger scale. And if an ideal dosage can be found that increases the lifespan of C. elegans, it would be worthwhile to test per tandem on other animal subjects, such as mice. Future studies could also look at gene expression to see if skin 1 gets upregulated in C. elegans the way that nerve 2 is supposed to in humans. Now, let us consider some conflicting evidence. So here's the thing. Both oxidants and antioxidants are naturally present in our bodies, whether we supplement or not. Oxidants create free radicals that help to kill pathogens and remove damaged cells, including cancer cells. But we also know that radicals can rip apart DNA and cause cells to become cancerous. In a study at McGill University, roundworms were bred that overproduce superoxides, and those worms live 32% longer than normal worms. So it could be an indication that superoxides may not necessarily be destructive molecules as so much as protective signaling molecules that turn up the expression of genes that help repair damaged cells.
This is very important because it questions the line of thinking and almost a movement that do calorie restriction to prolong life, because
That works.
Calorie restriction.
calorie restriction works.
They've done it in heavy, but it's not necessarily. It's not.
Different mechanism.
It doesn't have to be superoxides. It doesn't have to be ROS.
Yeah,
process.
right.
It might be
could
something else.
others.
And it's probably it's probably the insulin pathway. I can't remember now. Where
That's correct.
Yeah, it's insulin growth factor 1.
Yep.
And,
It's not a superoxide. It's
through,
not a ROS.
through PI3 kinase and AKT signaling. Yeah.
Similarly, worms bathed in a free radical promoting herbicide lived 58% longer than untreated worms. And in humans, it goes to, I think, maybe the same study you were talking about earlier, Dr. Fares. In humans, beta-carotene and retinol supplementation is linked to increased risk of lung cancer in heavy smokers.
They'd seen that, and then they did the study.
And this increased risk was more pronounced if the smokers were exposed to asbestos. It was a 1996 study of 18,000 people, so a very large study. And in a vigorous scientific review in the Journal of the American Medical Association, there was a higher rate of mortality among some antioxidant users. They looked at 68 randomized trials and with over 200,000 participants. What they found was a 5% increase in the rate of early death. And this was linked to beta-carotene, vitamin A, and vitamin E. So not all the vitamins.
Well, strictly because of vitaminosis?
I think it was probably, megadoses was probably a factor. I mean, the dosages range for all the participants.
Well,
but the mean dosage, it seemed to me, was pretty high.
But how much does your body absorb? I think there's a limit to your absorption, and there's a new study that shows that most of it goes into PP anyway. So it's not, you're not absorbing all of it, but it is ROS. What she's showing there, and what's saying, ROS is good. You need this response in your cells.
Of course, it's
That your
calibrated.
and if you block it, if you add too much antioxidants, if you completely block it, its cells don't respond to stress as well. And at least in the paper she's going to send you, the one I talked about, that one is p53 mediated. Because when you have the oxidized species, get DNA damage presumably, and then that activates p53, which stops cells. Cells, repair before you go on or die.
Yeah.
And if you don't have that, p53 is not activated. And you don't get to stop. You just keep going. You keep dividing even though you might have damage.
Yeah, I think balance is key.
Yes.
Yeah, yin
Down
and
with
yang.
potentium. Oh, sorry. We shouldn't say that.
Yin and yang.
So I'd like to end by discussing the interesting case of the naked mole rat. And I have a picture of one here.
She's
He's a
cute.
handsome little guy. What's interesting about him is that he exhibits a high level of oxidative stress. And yet he lives 8 times as long as a similar sized mouse, a typical lab mouse. And he has 6 times the amount of NERV2 expression.
6 times?
6 times the amount of NERV2 expression.
That is also something important to know, is that the amount of active NERV2 is not proportional to how much you express it, because I think mostly it's just floating around the cytosol, tied to KEAP2 in an unuseful way. And then when you separate it from that, then it migrates to the nucleus and it becomes to work. So
Yeah, we don't really know how much is tied up and how much is active.
yes,
Right.
so expression I think does not equal the actual use of it.
That's a good point.
I think that NERV2 translocation, the meaning of NERV2 translocation is not very well understood, to the point that you have a bunch of pharmaceutical companies looking in small molecules to see what they do.
Because it also turns out that it's not only enough to separate it from KEAP, but you have to build it. It first must accumulate, and then it migrates. So
But that makes the activation faster. That'll zoom at it.
Uh-huh.
If you have like 8 times.
Yeah.
Oh yeah,
That
but
does.
that probably. But for example, if there's a feedback loop and what is happening is that you're producing perfectly fine, but you just are not using it, or you cannot.
There's no way you're not using it. There's absolutely
cannot
zero
ubiquitous-
possibility that you're making 8 times the amount at steady
you're not
state,
using
and
it.
you're not using it.
Will
It's
be
a little bit like,
counterintuitive how
alpha
nature
methane.
works.
Yeah, I mean evolutionary. I mean,
Yeah.
I'm imagining. I'm the way I see it. It's almost
Yeah,
impossible.
I'm not an expert.
Either it's doing something already, which I bet you it
Next
is,
mole rat.
or it means that you can respond faster, much faster than anybody before. You build up levels and before, so DNA damage immediately is snuffed up, for example, or anything. Maybe,
Mhm.
but you don't have a supply of 8 times the amount.
What is known is that
I think.
if you give protandom in theory to patients with cancer, it protects cells that patients that are receiving chemotherapy, but you're also protecting tumoral cells, because the protandom doesn't discern, the.
So in the cancer models, as I said in the paper, in the postdoc, in the cancer model, when they add antioxidants, if you are P53 positive, if I remember correctly, the antioxidants are bad for you. And if you're P53 negative tumor, they don't affect you. So it links on P53. So they tested different tumors, and it falls if you're positive, if you're a cancer that you express P53, or it's the opposite. I can't remember now. But let's say if you express P53 and you add antioxidants, the cancer cells are proliferating much faster. But if you don't have 53, it doesn't matter whether you add antioxidants or not. So that seems to be the checkpoint, the link, the place where you.
Well, thank you for adding that, because I didn't know.
But you'd add potassium to these mole rats and see if you can kill them.
Or they turn yellow.
They're there for sure.
Their throats turn yellow.
They grow hair.
Another thing is that these naked mole rats, they have relatively short telomeres. And if you're familiar with the telomere idea behind aging, our chromosomes, or chromatids rather, have these repeating sequences at the end that portion gets chopped off with each subsequent cell division, so that the telomere gets shorter and shorter and shorter, until eventually that could lead to it could disrupt the integrity of the DNA. And the fact that naked mole rats have short telomeres kind of throws that theory into question as well.
Well, because it's not just the length of the telomere, it's also the telomerase activity, that is the enzyme implicated in shortening the telomere.
So mice are very similar, I think. Mice have long telomeres, but the size of their telomeres doesn't get shorter with age, just like in humans.
Right.
Right? So
And there's a difference between somatic cells and germ cells.
Yeah, well,
it's not a matter of
stem
how
cells,
short, it's a matter of do they shorten.
Yeah.
Because every time
They
I
might
do, I
not
think
shorten
they
in
shorten
age.
and then they get a little elongated, right? It's just that
Each
they
division
don't
you
grow as
shorten.
much as they get shortened on each cycle. Too,
Which,
Yeah, I'm not sure
with
how.
each division you
but our
shorten.
telomerase levels goes down, so every time you divide, the cells are dividing, of course.
I think.
You know, skin, bone marrow.
The
They are
method.
time their telomere length.
Yeah.
Every division.
So I work with Susan Price. They were expert on telomerase activity, telomere length, and the method used when I was in Cincinnati, the method used to look at telomere shortening are not so accurate. So because you can do it with microscopy or other methods, and all kind of by comparison. So there is not so accurate the tools that we have to measure that.
Okay, good to know. So the naked mole rat also has more ribosomes and more proteasomes, which are protein-making and protein-disposing machines. And this means they have a higher turnover of proteins, including the damaged proteins. And that could be a factor in why they have a high protein transcription accuracy. That's
How did
key.
you find out the story of the naked mole rat?
It was in several articles, so I just I sifted and read, and yeah, it's fairly recent. They're becoming more and more of an interest in terms of like aging studies because of their unusual longevity compared to other similar sized rodents. So this high protein transcription accuracy may be one of the reasons that they're also unusually resistant to cancer. But another proposed mechanism of their cancer resistance is in their cell sensitivity to each other. Their cells will stop dividing at a third the density mouse cells do. And we know that cancer cells don't stop dividing. They don't respond to those signals. And their cells are very sensitive to those contact signals. And it's because of this compound called hyaluronin, which makes their skin stretchy. We assume that's maybe the original purpose of that compound. And they need their skins to be stretchy and elastic when they're crawling through tight spaces so that their skins don't rupture. And then this side effect, the fact that hyaluronin perhaps confers cancer resistance, may be just a bonus.
Hyaluronic acid is a component of the extracellular matrix, and there is a high rate of turnover. So we used to measure hyaluronic acid in the effluent of livers that were harvested to see if the liver were good or not good. So it's a component of cell regeneration. It's not just only applied to the naked mole, but it's interesting.
Yeah. And there's not only one way to defend against cancer, because blind mole rats are also resistant to cancer, but they do it through a different method by commit suicide signals to rapidly growing cells. Was I on this one the whole time? I'm sorry. Okay. So, in conclusion, given the conflicting evidence against the oxidative damage theory, perhaps it's time to revise it or propose a new one that takes into account the balance required between oxidants and antioxidants in the chemical reactions that are necessary for normal aerobic life, and perhaps a shift from the oxidative management theory. And that's it. So at this point, I can take any questions or comments that you